Meet Marcus Brown, a talented football player who recently found himself at the center of a medical mystery. He's a high school senior and the leading scorer on the team, but has a history of feeling faint and passing out towards the end of second practice, thought to have been due to dehydration or having just gotten over a bad cold. On the third game of the season in August, despite the weather being very hot and humid, he was feeling well on game day, and his team was relying heavily on the passing game. By the end of the first quarter, Marcus had already run 18 pass routes and had 5 receptions, as well as 1 touchdown. However, by the last quarter, as he was jogging back to the huddle, he suddenly staggered and collapsed. The coach and trainer ran to him and noted he was not breathing and had no pulse. After 1 to 2 minutes of CPR, a weak pulse was detected and he began to breathe on his own, before EMS arrived and rushed him to the nearest hospital. So, what happened to Marcus? In order to find out, we'll cover the specialized parts of the cell membrane, how interactions, biomolecules, and cell transport, and try these concepts into Marcus's case throughout the video before tying everything together at the end. Exhibit 1. The cell membrane. The guardian of every living cell. This is how scientists describes what the cell membrane looks and functions like, because it's made up of different molecules that are distributed across the cell membrane. The movement of the mosaic of molecules makes it impossible, however, to form a completely impenetrable barrier. This is where selective permeability comes in. Now, the role of the cell membrane is to maintain this selective permeability, meaning it controls the entry of substances into and out of the cell. This allows essential molecules to enter while keeping out harmful substances, helping to maintain the cell's internal environment, or homeostasis. Now, in this case, carbohydrates like glucose use carrier proteins to traverse the membrane. During Marcus's football game, dehydration led to the reduced hydration of the bilayer, causing the phospholipid tails to become tightly packed. This change affected the lateral movement of integral proteins, which is responsible for ion transport, potentially causing muscle weakness and collapse. Let's bring it back to the basics. Exhibit 2. Hydrointeractions. Now, there are 2 very important parts of a phospholipid, the head and the 2 tails. The head is a phosphate molecule that's attracted to water, or is hydrophilic. 2 tails are made up of fatty acids, or chains of carbon atoms that aren't compatible with or repel water, which are hydrophobic. The head contains a phosphate group and is polar, while the tails are composed of fatty acid chains that are nonpolar. In the cell membrane, phospholipids arrange themselves into bilayer, with the hydrophilic heads facing the surrounding water and the hydrophobic tails facing away from the water. This is also what makes cell membrane semi-permeable, where the hydrophobic tails in the middle make it difficult for water-soluble or polar molecules like ions and sugars to pass through. Now, fatty acids are what makes up phospholipid tails. Saturated fatty acids are chains of carbon atoms that have only single bonds between them. As a result, the chains are straight and easy to pack tightly. Unsaturated fats are chains of carbon atoms that have double bonds between some of the carbons. They play a role in membrane fluidity because they increase the space between the phospholipids, making the molecules harder to freeze at lower temperatures. Now, simple diffusion allows nonpolar molecules to move through the lipid bilayer freely, driven by concentration gradients. Facilitated diffusion, on the other hand, involves integral membrane proteins assisting the transport of hydrophilic or polar molecules. Now, dehydration changes in membrane fluidity can slow down diffusion, affecting the transport of various substances, and therefore causing Marcus to pass out. Dehydration can lead to elevated electrolyte concentrations in the blood, such as sodium. This is also called hypertonic dehydration, which is the imbalance of water and sodium in the body. The altered fluidity of the bilayer may have impacted the efficiency of these proteins, leading to Marcus weakness and Marcus. The cell is made up of 2 different types or classes of proteins. Integral proteins are nestled into the phospholipid bilayer and stick out on either end, while integral proteins are helpful for transporting larger molecules like glucose across the cell membrane. Polar means that the electrons are not evenly distributed, making one side of the molecule more positively charged or negatively charged than the other side, while nonpolar means that the electrons are evenly distributed, so the molecule is evenly charged across the surface. Now, phospholipids are attracted to each other, but they're also constantly in motion and bounce around, creating small spaces that make them semi-permeable. One way of distinguishing between what can pass through and is based on how they react with water. Now, small polar and nonpolar molecules and large nonpolar molecules, that's confusing, like oxygen, carbon dioxide, water, and carbon pass through the phospholipid bilayer with simple diffusion, but they may pass slowly due to their hydrophobic tails. Now, the size and charge of large polar molecules and ions make it too difficult to pass through the nonpolar region of the phospholipid membrane, without help from transport proteins. Now, in Marcus's case, dehydration leads to a drop in body water, disturbing the balance of vital ions like sodium and potassium, which are essential for cell function and osmotic balance. But dehydration can make it more rigid, affecting its selectivity and causing problems with ion movement. This disruption can lead to an accumulation of ions inside or outside the cell, disrupting its polarity. In Marcus's case, the disruptions in ion transport, particularly in nerve and muscle cells, can lead to depolarization, which can affect nerve signaling and muscle contractions. This is why he experiences muscle weakness and fatigue as a result of his dehydration. Exhibit 3. More biomolecules. First, integral proteins are channel and carrier proteins responsible for ion transport, signal transduction, and structure. Channel proteins are transmembrane protein that travels completely through the cell membrane. They move large or polar molecules and ions that cannot pass through the cell membrane on their own through facilitated diffusion. Now, carrier proteins are transmembrane proteins as well that have a binding site for large polar molecules and ions. And finally, peripheral proteins are found temporarily attached to integral proteins but are not embedded in it, and function and support communication, enzymes, and molecule transfer in the cell. Also, one important cell not shown here are receptor proteins, another transmembrane protein that has a bind that extends into the external environment for cell signaling, where a signaling molecule attaches to its receptor, and the protein changes shape and causes a change inside the cell. Now, cholesterol is another key player in maintaining the fluidity of the cell membrane. They act as a fluidity buffer, preventing the fatty acid chains in the phospholipids from packing too closely by inserting into phospholipids, and preventing them from interfering from each other to avoid aggregation at lower temperatures, which can make the membrane too rigid. In contrast, at higher temperatures, cholesterol increases fluidity and reduces the disorder caused by the excessive movement of phospholipids. Now, glycolipids play a crucial role in cell recognition and adhesion, which carbohydrate chains are identification markers that allow cells to distinguish between self and non-self, and recognize other cells, pathogens, or molecules. In terms of dehydration, the carbohydrate portion of glycolipids is hydrophilic, meaning it readily interacts with water. Dehydration can change the conformation and orientation of these carbohydrate chains. In some cases, severe dehydration can lead to denaturation and misfolding of these chains, which could affect their function in self-recognition and adhesion. Now, receptor proteins are essential for signal transduction passages in cells because they bind to specific signaling proteins or ligands. Now, ligands vary in nature from small hydrophobic molecules like steroid hormones to water cell-soluble ligands like peptide hormones and neurotransmitters, meaning the interaction between ligands and the receptor proteins is the initial step in cellular signaling. Now, in Marcus' case, the body relies on such specialized receptors to detect changes in its internal environment, such as a decrease in blood volume or an increase in blood osmolarity due to fluid loss, such as through stressing or inadequate water intake. When these changes are detected, specific receptor proteins like osmoreceptors, located in the hypothalamus of the brain, send signals to burst and release antidiuretic hormone, or ADH, from the pituitary gland. ADH acts on cell surface receptors in the kidneys, called D2 receptors, to increase water reabsorption, thereby reducing urine input and conserving water. In this case, a malfunction in Marcus' receptor-based signaling system likely contributed to its dehydration. Now, Exhibit 4, cell transport. Simple diffusion, which plays a role in the movement of small nonpolar molecules like oxygen through the cell membrane, ensures that molecules move from areas of higher concentration to lower concentration, which is essential for the intake of oxygen by Marcus' muscle cells. During diffusion, substances move from high to low concentration until the concentration becomes equal throughout space, called the concentration gradient. This is also true for some substances moving into and out of cells. Because the cell membrane is semi-permeable, only small uncharged substances like carbon dioxide and oxygen can easily diffuse across it. The middle of the cell membrane is hydrophobic, meaning only nonpolar molecules can pass easily through it as they're lipid soluble. Now, when Marcus is exercising and breathing healthily, the transport of oxygen into his cells mainly involves simple diffusion, where oxygen molecules, being small and nonpolar, can diffuse through the phospholipid bilayer in the cell membrane. This is especially important for Marcus's muscle cells, which require oxygen for energy production during exercise. Oxygen molecules move from an area of higher concentration in the alveoli of the lungs and in the bloodstream to an area of lower concentration inside his muscle cells through simple diffusion. Dehydration, the heightened solid concentration within the cells, causes an increase in osmotic pressure. This prompts water to move out of the cells into the bloodstream in an effort to restore balance. As a result, cells lose water, shrink, and increasing extracellular solute concentrations. And here the net movement of water is from hypotonic to hypertonic regions. In this case, cells become hypertonic due to the loss of water, while the extracellular environment is relatively hypotonic because it has a higher solute concentration. As a result, water moves from the hypotonic extracellular fluid into the hypertonic cells in an attempt to equalize the solute concentration, helping to rehydrate the cells. This means the cell has a high water potential. Now, in Marcus' case, water would move from the cells where the water potential was higher. The extracellular space where the water potential was lower due to the increased solute concentration. Now, this movement of water out of the cell results in lower pressure potential. As a result, Marcus may experience a drop in blood pressure during the osmosis, and an isotonic solution has the same solute concentration as the cell's cytoplasm, resulting in no net movement of water. Cells in isotonic solutions maintain their shape and size. In a hypotonic solution with a lower solute concentration than the cell, water moves into the cell, causing it to swell and in some cases burst. Conversely, a hypertonic solution has a higher solute concentration than the cell, causing water to move out of the cell. This leads to cell shrinkage or shriveling. Now, the IV bag given to Marcus likely contained a balanced electrolyte solution. In this case, IV bags are often used to deliver saline solutions, which are isotonic with human blood. An isotonic solution has the same concentration of solutes like salts as the intracellular and extracellular fluids. When an isotonic solution is administered intravenously, it helps maintain the balance of solutes between the bloodstream and the cells. On the other hand, facilitated diffusion plays a pivotal role in transporting larger polar molecules, like glucose, into cells. This process involves molecules or ions moving across cell membranes with the assistance of transport proteins. Now, dehydration raises solute levels in Marcus' bloodstream, particularly in the extracellular fluid. This causes higher solute concentrations outside the cell compared to the inside. As a result, aquaporins move water out of the cells to balance concentrations in solute-rich extracellular fluid, while transport proteins aid in solute balance between the extracellular fluid and the cell interior. Now, there's three factors that may have caused Marcus to faint at dehydration and that influence how his body maintains homeostasis: the concentration gradient, surface area, and heat. One not mentioned here is the thickness of the exchange surface. If it's thick, then the molecules have bigger distance to diffuse across, slowing the rate of diffusion, while thinner exchange surfaces increase the rate of diffusion. Now, the transport of glucose molecules from Marcus' food into cells also involves facilitated diffusion, where glucose molecules are polar and relatively large, making it challenging for them to pass directly through the cell, because it has a hydrophobic interior. Now, to overcome this, glucose transport is facilitated by transport proteins. Now, all three modes of passage transport follow this concentration gradient to equalize the concentration of solutes on both sides of the membrane, meaning they don't require any energy. Now, active transport. During active transport, substances move against the concentration gradient from an area of low to high concentration. This process is active because it requires the use of energy in the form of ATP. It's the opposite of passive transport in this way. Now, as opposed to facilitated diffusion, active transport goes against the concentration gradient and uses carrier, not channel, proteins. Now, during dehydration, the loss of bodily fluids can lead to an increase in the concentration of electrolytes, particularly sodium in the extracellular fluid. Now, the sodium-potassium pump plays a crucial role in maintaining the resting potential of the cell's membrane and maintaining electrolyte balance by actively pumping sodium out of cells and importing potassium into cells to establish an electrochemical gradient. This process helps counteract electrolyte imbalance caused by dehydration, preserving cell function and preventing complications associated with excessive electrolyte concentration in the extracellular space. There are variations in endocytosis, but all follow the same basic process. First, the plasma membrane of the cell invaginates or folds inwards, forming a pocket around the target cell, target particle or particles. The pocket then pinches off with the help of specialized proteins, leaving the particle trapped in a newly created vessel or vacuole inside the cell. Cells must take in certain molecules such as nutrients, but they also need to release other molecules such as signaling proteins and waste products to the outside environment. Now, exocytosis, exo meaning external, cytosis equals transport, is a form of bulk transport in which materials are transported from the inside to the outside of the cell in membrane-bound vesicles that fuse with the plasma membrane. Now, dehydration often results in an increased concentration of waste products and solids from within the body. In response, cells may enhance exocytosis as a means to expel cells waste and secretions. However, dehydration can hinder this process due to the reduced availability of water, making it more challenging for cells to effectively package and eliminate waste materials. Consequently, cell byproducts may accumulate within the cell. Now, apart from concentration gradients, cells also maintain the membrane potential, typically between -40 and -80 millivolts, resulting from a net charge separation across the cell membrane. This electrical potential difference, also called membrane potential, is actively upheld. For example, in the case of sodium and potassium ions, the movement is determined by both concentration gradients and the membrane potential, collectively known as electrochemical. Now, secondary active transport uses the energy stored in these gradients to move other substances against their own gradient. As an example, let's suppose we have a high concentration of sodium ions in the extracellular space, thanks to the hard work of the sodium-potassium pump. If a route such as a channel or carrier protein is open, sodium ions will move down their concentration gradient and return to the interior of the cell. And finally, Exhibit 5. Dehydration can alter the fluidity and permeability of the phospholipid bilayer. As water is lost from the cell, the lipid molecules in the bilayer may become more closely packed together, making the membrane less fluid, more rigid, and less permeable to water, ions, and large molecules. In response to dehydration, the cell may try to reduce water loss by decreasing membrane permeability, thus conserving intracellular water. Now, cholesterol also helps to maintain the fluidity of the cell membrane by preventing the phospholipid molecules from packing too closely together, recall fatty acids. Dehydration can lead to changes in cholesterol distribution within the membrane, also affecting its stability. Now, phospholipids in the cell membrane have a structure with hydrophilic or water-trapping heads and hydrophobic water-repelling tails. Dehydration, which involves the loss of water from the cell, leads to changes in the arrangement of these phospholipids. As the cell loses water, the hydrophobic tails can come closer together. This change in the phospholipid structure makes the membrane less fluid and more rigid. The close packing of the hydrophobic tails reduces membrane permeability, preventing excessive water loss. This also means that the sodium and other ions, which typically rely on transport proteins, can also be impeded, affecting nerve signaling, muscle contraction, and osmotic balance. As a result, they would use ion channels and transporters to try and circumvent this. Now, dehydration often leads to an increase in the osmolarity or concentration of solutes of the extracellular fluid. Cells need to sense this change and respond to it to maintain osmotic balance. Receptor proteins on the cell membrane, such as osmoreceptors, can do this, and they detect the increased osmolarity and trigger signaling pathways that lead to the release of antidiuretic hormone, or ADH. These hormones help the body retain water by increasing water reabsorption in the kidneys, which ultimately helps counteract dehydration. Receptor proteins also trigger the feeling of thirst and stress hormones. Now, dehydration also affects osmosis by creating a concentration gradient that drives the passive movement of water out of cells. As a result, the solute concentration in the extracellular fluid decreases. This higher solute concentration outside the cells creates an osmotic pressure, causing water to move out of the cells through osmosis in an attempt to equalize the concentrations, and dehydrate, leading to cell shrinkage. And finally, active transport. In the sodium-potassium pump, dehydration can lead to an increase in the concentration of sodium ions in the extracellular fluid, as a loss of water through sweating or urination. This increase in extracellular sodium levels can affect the sodium-potassium pump's ability to maintain the proper sodium and potassium concentration gradients. Dehydration can also lead to reduced ATP availability, which can impair the pump's ability to maintain the proper ion gradients. As cells shrink, the intracellular sodium and potassium concentrations can become imbalanced, leading to changes to the resting membrane potential and an increased risk of membrane depolarization, which can result in decreased nerve and muscle function as well. So, why should you care about what you drink? What you choose to drink not only affects your hydration, but also the balance of electrolytes and the efficiency of transport proteins and processes like the sodium-potassium pump, which helps regulate cell membrane potential, osmosis as well. Now, beyond all of these processes and transport modes, we have just a general overview of why should we care. The proper hydration supports these cell functions, ensuring effective nutrient exchange and waste removal, gas exchange, joint and organ protection, regulation of temperature, and so much more. As they say, the human body is 60% water, and what we've covered here is a testament to how that 60% is that important to your lifestyle. Thank you.